In this class series, we are discussing about the grade 12 plus NEET past paper questions, including the important topics from the particular chapters as well. So today we have the chapter Biomolecules. First, let us go through the important topics from the chapter. Then we will move to the questions. Okay, so let's begin from the chapter, that is biomolecules. So first of all, we have to know what is biomolecules. Biomolecules means the compounds or the molecules which are present in our human body. And here we have a different kind of all the chemicals present in our body. They are biomolecules, right? Vitamins, carbohydrates, proteins, all of them are biomolecules. So here we have to know the structure of these compounds, what are the functions of these compounds and all those things. Okay. So biomolecules, actually we have a sequence in our body, right? Biomolecules are combining together to produce cells, which combines together are combining to produce the overall living system. So this is the human body actually. So now first we have to know about the carbohydrates. Actually the carbohydrates are produced by the plants and they will form very large group of naturally occurring compounds. So the carbohydrates are usually including cane sugar, glucose, starch and everything. And coming to the general formula, that is C6H2O by times, and they can be considered as hydrates of carbon. Okay, so suppose if you consider glucose, which has in the molecular formula C6H12O6, we can write it as C6H2O 6 times. So this is the carbohydrate structure, that is fructose has the given structure, that is hydroxyl group is there, keto group is there. Okay, it means these carbohydrates are very, very important to our body. Okay, now but the thing is all the compounds cannot be considered as the carbohydrates. There are some other compounds also having the same formula, that is acetic acid, but which is not a carbohydrate. Okay, but commonly we can say that's it. Now coming to the carbohydrates, they are actually optically active polyhydroxyaldehydes or ketones, and they are also called saccharides. You might have heard about monosaccharides, disaccharides, right? So these are also called saccharides. And if you consider the structure of glucose and fructose, you can see from this diagram, in glucose there is no keto group, there is only aldehyde group. While in fructose, there is a keto group. So these all are examples for carbohydrates. Okay. Now classification of hydrocarbons. So as we discussed now, hydrocarbons are also, classification of the carbohydrate. So as we discussed now, they are also called saccharides. So they can be classified into monosaccharides, oligosaccharides and polysaccharides. Okay, so this is completely based on their behavior on hydrolysis. Okay, so first one is monosaccharides. Monosaccharide means the carbohydrate which cannot be hydrolyzed further to give simpler units are called monosaccharides. We have around 20 monosaccharides are there, and this includes glucose, fructose, ribose, etc. So monosaccharides means we cannot split them again. That is monosaccharide. Okay. Now coming to the oligosaccharides, the carbohydrates on hydrolysis will produce us 2 to 10 molecules of monosaccharide units. Means on the hydrolysis they can produce around 10. Means not exactly 10, 2 to 10 number of monosaccharides can be produced from the carbohydrate. Those kind of carbohydrates are called oligosaccharides. Okay, and these oligosaccharides can be again classified into disaccharides, trisaccharides, tetrasaccharides and etc. Okay, so this is the classification. So depending on the number of means, if they are producing two monosaccharides, it will be disaccharide. If the oligosaccharide is producing 3 units, they can be called as trisaccharides. If they are producing 4 units, then there will be tetrasaccharides and so on. So based on the number of monomeric unit, means number of monosaccharides they are producing, we can classify them as these mono, di, tri, etc. So among these different kind of oligosaccharides, the common one will be the disaccharides. Okay, the disaccharides means they will be containing two monosaccharide units. For example, sucrose. Sucrose is a disaccharide because when they are doing we are doing hydrolysis of the sucrose, they will produce one molecule of glucose and one molecule of fructose. Another one, if you consider maltose, they can produce two molecules of glucose. So this also one or two examples are here. So we have to know other examples as well, because in NEET exam they can ask those kind of questions. Next one is polysaccharides. So polysaccharides means the carbohydrates are producing more number of monosaccharide units. Okay, if it is 2, we call it as 2 to 10, we called it as oligosaccharides, right? So the carbohydrates which is producing large number of monosaccharide units are commonly called polysaccharides. Okay, so examples we can say starch, cellulose, glycogen, gums, they are examples for polysaccharides. Okay, so this for example, starch on undergoing hydrolysis, they can produce n number of glucose molecules. That is an example. And the another thing is the polysaccharides are actually not sweet in taste, and they can be called as non-sugars. Okay, for example, this glucose starch is not that much sweet. It means they are not at all sweet in taste, but they can be, so they can be called as non-sugars. And they are amorphous in nature, then they will be insoluble in water. That is another thing regarding the polysaccharides. So this is the summary of the classification: monosaccharide, oligosaccharides, polysaccharides. Monosaccharides cannot be hydrolyzed further. Oligosaccharides can yield 2 to 10 molecules of monosaccharides. Polysaccharides yield a large number of monosaccharide units on hydrolysis. Next one, this is the classification based on the number of monosaccharides they are producing during the hydrolysis, right? Now we are going to the classification on the basis of their nature. So they are of two types: reducing sugars and non-reducing sugars. So what is a reducing sugar? Reducing sugars means the carbohydrates which will reduce the filling solution and tolerance reagent. Okay, so the carbohydrates which can reduce the filling solution and tolerance reagent are commonly called as reducing sugars. And all monosaccharides, whether aldose or ketose, are reducing sugars. So this is completely based on the chemical reactivity. So the carbohydrates that can reduce the filling solution and tolerance reagents are called reducing sugars. Now coming to the non-reducing sugars, the carbohydrates that cannot reduce the filling solution and tolerance reagents are called non-reducing sugars. So reducing sugars will be reducing the tolerance reagent, and non-reducing sugars cannot reduce the filling solutions or tolerance reagent. So based on this reaction only we can classify them. That is another thing. Okay. Now for example, glucose and fructose are reducing sugar. Sucrose is a non-reducing sugar. So why they are reducing sugar? Because in the reducing sugars, there will be aldehydic or ketonic group. But in case of non-reducing sugars, there is no any free aldehydic or ketonic. That should be free. So in reducing sugar, free aldehydic or ketonic group will be there. But in non-reducing sugars, it is absent. Now we are moving to the classification of the monosaccharides. How can we? So we know what are monosaccharides. They means they will undergo hydrolysis and produces no other monosaccharides. No other units can be produced, right? So classification of monosaccharides means we can be classified them into aldose sugars and ketose sugars. So aldose sugars means they will be usually containing aldehyde group, while the keto sugars means they will contain the keto group. So if it is a triose, if the number of carbon atoms are 3 and they are containing the aldehyde group, that will be aldotriose. If it is number of carbon atoms still 3 and it is containing keto group, it will be ketotriose. So this is the classification. Similarly, if it is 4 carbon compound and containing the aldehyde group, it is aldotetrose. And if it is containing keto group, it will be ketotetrose and so on. That is the classification. So like this, we can classify them in different way. Okay. Now we have to know the preparation and structure. Usually the glucose will be a colorless crystalline solid and they are soluble in water and they will be sweet in taste. So here we can see the structure of the glucose. They are containing the aldehyde group and hydroxyl groups are also present. But here this carbon is having the different configuration. Other carbon atoms are having the same configuration, means the hydroxyl group on the same side. So this is the structure of glucose. Now we have to know what is the preparation of glucose. So glucose is a monosaccharide, right? So how can we prepare it? So first method means they can be prepared. Actually, they will be free in nature as well as in the combined form. So they are usually present in all sweet fruits and honey and everything. So here we have to know the preparation from sucrose and starch. From the sucrose we can produced by the hydrolysis, acid hydrolysis, and from a starch also we can obtain the by using sulfuric acid at a temperature of 393 Kelvin. So these are the preparation. Now we are moving to the structure. In the structure of glucose also we can see they have, actually glucose is an aldohexose and it is also known as dextrose because it is containing the aldehyde group and 6 carbon atoms. So number of 6 carbon atom, number of carbon atoms 6 means it will be a hexose sugar, and aldehyde group is there means it is an aldohexose. That is the classification. Now the structure of glucose based on the evidences. Now we know we have to know the cyclic structure of glucose. Actually, they can exist in alpha form and beta form. In the alpha form, which is obtained by the crystallization from concentrated solution of glucose, and beta form can be obtained by the crystallization from hot and saturated aqueous solution at 371 Kelvin temperature. So these two structures we have to memorize because in the questions you can expect those kind of questions. Okay, exams. So these structures are also very, very important. Now let us consider the process mutarotation. So mutarotation means it is the change in the optical rotation because of the change in the equilibrium between two anomers. Okay, when the corresponding stereo centers interconvert, cyclic sugars show mutarotation as alpha beta means this is the process of mutarotation, that is the change in optical rotation because of the change in the equilibrium between two anomers. That is the thing. Now here we have to know the structure of fructose as well. So the structure of fructose means it is actually obtained from the structure of furan, and these cyclic structure as well as these two structures should be memorized. Next is in case of disaccharides. So disaccharides means we know there will be two monosaccharide units, right? So there will be, it is usually joined by an oxide linkage produced by the loss of water molecule. Such a linkage can be called as glycosidic linkage. So in case of disaccharides, the type of linkage present is called glycosidic linkage. That is also important. And there will be a C1 of alpha and C4 or C1 C4, that is the linkage will be C1 C4 glycosidic linkage. So that is the name of the linkage is also important. Okay, now the polysaccharides. So we know the polysaccharides are containing a large number of monosaccharide units, right? They are joined together by the glycosidic linkages, and they can be mainly act as the food storage or structural mineral. That is also important. So this is the main properties of this one. So we have to know the examples for the polysaccharides. The first one is starch. So starch means it is a storage polysaccharide of plants, okay, and it is the most important dietary source for human beings, and high content of the starch is found in cereals, roots, tubers and some vegetables. and they are usually containing, when it means that it is a polymer of glucose, alpha glucose, and they are of containing two components. They are amylose and amylopectin. If you consider amylose, yeah, this amylose and amylopectin, they are having different properties. Actually, amylose are usually water soluble component, and they are contributing 15 to 20 percentage of starch. At the same time, if you check the structure, this amylose is a long unbranched chain with 200 to 1000 number of D-glucose units held together by C1C4 glycosidic linkage. So the structure of amylose is also important. The important difference is amylose is water soluble, but amylopectin will be water insoluble, and it constitutes about 80 to 85 percentage of the starch. Okay, and it is a branched chain polymer of alpha D glucose. Before, amylose was the unbranched chain polymer, but this amylopectin is the branched chain polymer of alpha D glucose. That is another important thing, and they are connected by C1C4 glycosidic linkage, and the branching occurs by C1C6 glycosidic linkage. Means the type of linkages also we have to memorize, because the questions are like that only. Okay, so we can see the questions like some similar questions in the previous past papers. Okay, then this is the structure of starch. This is the complicated structure, but still we have to know this. Now coming to the structure of cellulose. Cellulose is another kind of polysaccharide, right? So they are cellulose occurs exclusively in plants, we know that, and they are most abundant organic substances in plant kingdom. And coming to the cellulose, it is a straight chain polysaccharide composed only of beta D glucose units, which are joined together by glycosidic linkage between C1 of one glucose unit and C4 of the next glucose unit. So here the linkage is C1 C4 linkage, and the structure is also important. Now coming to the third one, that is glycogen. Glycogen is the carbohydrates that is stored in the animal body. Usually the glycogen form is obtained or can be seen in case of animal body. Plant body it is another. So that is the different things, and this structure is similar to that of the amylopectin. So these are the important points, and glycogen is also found in yeast and fungi. So this is also important. Now, this is all about carbohydrates. Now we are moving to the proteins. So proteins means we know they are most abundant biomolecules in the living system, right? So in case of proteins, we know there are different sources of proteins that includes milk, cheese, pulses, peanuts, fish, meat, and etc. This is very important for our body, right? And now they occurs in every part of the body and form the fundamental basis of the structure and functions of life, and they are required for the growth and maintenance of the body. And word protein is okay, that is not important, but here we have to know what are the important examples of proteins and what are the rules, means what are the roles of protein in our body. So actually all the proteins are it is a polymer composed of the monomers amino acids. So all proteins means amino acids are the building blocks of proteins. So proteins are polymers of alpha amino acids. Okay, so if you consider amino acid, we can see the functional groups, that is amino group and the carboxylic group. So both these functional groups are present in the amino acid. These kind of amino acid units will combine together to produce one protein chain. Okay, so now coming to the amino acids, they are both containing both the groups as we discussed now, NH2 group and COOH group. And depending upon the relative position of the amino group with respect to the carboxyl group, the amino acids can be classified as alpha, beta, gamma, and delta, like different kind. Okay, only alpha amino acids are obtained from hydrolysis of proteins. That is also important. So they are usually containing other functional groups as well. Okay, so all alpha amino acids have trivial names which usually reflect the property. So depending on the position of the amino group, we are classifying the amino acids, right? And different amino acids are having different names as well. For example, glycine. Glycine is so named since it has a sweet taste. And tyrosine, it is first obtained from cheese. That's why, means different amino acids are having different names, and there is a reason as well. So amino acids can be usually classified into two, that is acidic amino acids and basic amino acids. So the acidic amino acids examples we can say aspartic acid, glutamic acid, and basic amino acids will be lysine, arginine, then histidine. It means these are the different examples of amino acids. Okay. Now the next one, classification. The amino acids can be classified into different, that is non-essential amino acids and essential amino acids. So non-essential amino acids means we are not requiring these. Anyway, the proteins or amino acids are important for our body, but the non-essential amino acids means they can be synthesized in our body and doesn't require from other external source. So the amino acids that can be synthesized in our body are called non-essential amino acids, and that cannot be synthesized are called essential amino acids. So here, as I told you before, we have different types of amino acids, and the examples of different type of amino acids are here. Essentially include histidine, isoleucine, leucine, lysine, those examples. Then non-essential include alanine, asparagine, aspartate, glutamate, serine, means different examples are there. So this is the classification. So we can classify them into neutral, acidic, basic, and essential and non-essential as well. How we are classifying them into neutral, acidic, and basic? As we discussed before, the amino acids are containing both NH2 group and carboxylic group, right? If the number of carboxylic groups and the NH2 groups are same, we can call it as they are neutral, because NH2 group is usually basic group. It is basic in nature. The carboxylic group means they are acidic. So if the compound, if the amino acids are containing equal number of both the groups, the total effect will be neutral. So they are neutral amino acids. If they are containing more acidic groups, they means more carboxylic group, they will be acidic. If they are containing more basic groups, they will be, means amino groups, they will be basic in nature. This is the another classification. The next one is the structure of protein. As we discussed before, there is a group, a carboxylic group of amino group. So the bond between, so amino acids are combining together to produce the protein, right? So when two amino acids are combining, they will release one water molecule, and the bond produced is called peptide bond or peptide linkage. Okay, so this peptide bond is the reason for the formation of the amino protein molecule. So this peptide linkage, see, from COOH we will release OH group, and from NH2 we can release one hydrogen. So I will show the removal. From the OH group we can COOH, we can remove the OH group, and from NH2 we are removing one hydrogen. So the remaining will be CONH linkage. So this linkage is called peptide bond, peptide linkage, or amide linkage. So three names are there: amide linkage, amide bond, peptide linkage, amide or peptide bond. Okay. So this is the classification. Another classification is based on their molecular shape, that is fibrous proteins and globular proteins. So in fibrous proteins, the polypeptide chains run parallel and are held together by hydrogen and disulfide bonds. But in case of globular protein, they will be, the polypeptide will be coiled around to give the spherical shape. That is the difference. And we have to know the examples. Fibrous proteins includes keratin, myosin, and in case of globular protein, examples are insulin and albumins. So some examples also we have to remember. And the proteins, they have different structures as well, that is primary structures. So primary structures means, see, we can we can see, we can understand the difference of these different kind of structure by using a diagram that I will show you. Before that, the proteins may have one or more polypeptide chains, right? So each polypeptide chain in a protein has amino acid linked with each other in a specific sequence, and it is the sequence of amino acids that is said to be primary. So the single chain is called primary, because a protein means it will contains many polypeptide chains. So the single chain is called the primary structure. So the primary structures are usually combining together to produce the secondary structure. So secondary structure means the one polypeptide chain, they can means the two they will coil together to produce the secondary structure, and they can coil in two ways. First one is alpha helix or beta pleated. So whatever the however they are doing that, that is no reason, but this is called the secondary structure. So these two, when these two strands are combining together, there should be some connection between these two strands. That is possible by the formation of hydrogen bond. Now, this is the beta structure, that is that hydrogen bond will be intermolecular, right? Now coming to the tertiary structure. Tertiary structure means this is the overall folding of the polypeptide chain. They will fold again and produce the tertiary structure. And here we can see the diagram as well. And coming to the quaternary structure, it means many more polypeptide chains will combine together. See, here we can see it is a complicated structure. Okay, so here we can identify the primary, secondary, tertiary, quaternary structure. See, the single strand is called primary, and they coils itself is producing the secondary, and two different kind of primary structures are combining to produce the tertiary, and quaternary means many of them will combine together. So these structures are also important. Okay. Next is the denaturation of protein. Denaturation of protein means it is simply the losing the activity of the protein. If the protein molecules, different protein molecules are having different functions in our body. So if they are losing their function, that is called denaturation of the protein. Okay. So example, the coagulation of the egg white on boiling, curdling of the milk which is caused due to the formation of lactic acid, means these are entirely changing the properties of the protein. So those processes are called denaturation. Now we are moving to the enzymes. Enzymes means they are the compounds or biological molecules that significantly speed up the rate of chemical reaction takes place within the cells. We know what are catalysts, right? They can increase the rate of the reaction without participating in it. So enzymes are actually, they are biological catalyst, means they can enhance the rate of the reactions which takes place in human body. That is called enzymes. Okay. So enzymes are also different types. Different enzymes are having different functions. For example, the hydrolysis of maltose can be enhanced by the enzyme maltase. So there are different types of enzymes. Okay. So if you come to the enzyme reaction, that is also different. It is interesting. Here we have the enzyme. So these are the substrates. So how the enzymes are speeding up the process. See, the enzyme, the substrates are usually reacting with the enzymes, and these substrates will be fixed in the active site. The enzymes are having some active sites, right? So the substrates will fix in the active sites of the enzyme, and there will be a formation of enzyme substrate complex. So this enzyme substrate complex will be then converting into the product, means on the surface of the enzyme, the substrate, means the reactants will react together, and after the product formation they will dissolve from the surface of the enzyme, and the enzyme will be regenerated. So here you can see that there is no any change in the shape or properties of the enzymes. They are actually providing the surface for the formation of the products. Okay, now coming to the vitamins. Vitamins are also essential for our body. They are complex organic molecules which cannot be produced by the body and must be supplied in small amounts in diet to carry out essential metabolic reactions which are required for normal growth and maintenance of the body. Actually, the vitamins are not synthesizing in our body. It is obtaining from our diet. Okay, so there are different types, means first one is water soluble vitamins and another one is fat soluble vitamins. So water soluble vitamins means they are soluble in water and they are supplied regularly in diet as they are regularly excreted in urine. That is the thing. Because water soluble vitamins means they can be excreted from our body through urine, right? So we have to provide a regular supply of these vitamins to our body, and these are including the examples vitamin B1, B2, B6, B12 and C. Now coming to the fat soluble vitamins, they are usually soluble in fat and oils, and they are usually stored in liver and adipose tissues. So this is the classification, and here the examples we have to memorize. So all the B complexes means B1, B2, B6, B12 are water solubles, and C is also water soluble. That is, we can see in lemon, right? So fat soluble vitamins means they will be A, D, E, K. So these examples also we have to name. And here we have some important sources and sources of different vitamins. And here we have mentioned if the deficiency of vitamin happened, what it will leads to what kind of situation. So the deficiency of different vitamins are leading to different diseases. That is deficiency diseases. So that is also given here. Okay. Vitamin A is usually obtaining from milk, butter, egg yolk, green and yellow vegetables, etc. So if the deficiency is there, it will cause different kind of diseases such as night blindness, xerothalmia, those kind of. Okay. So vitamin B1 is also important. The deficiency will produce beriberi, and vitamin B2 will produce cracked lips, sore tongue, digestive disorders, and burning sensation of the skin. Vitamin B6 is also producing nervous disturbance. Vitamin B12 is producing pernicious anemia or RBC deficit in hemoglobin. So here this is important because in the previous questions we have seen that there are some questions from these vitamins as well. So this is also very important topic. Okay, and vitamin C, D, E. So all of the vitamin deficiencies are creating different kinds of disorders in human body. And next is the nucleic acids. We have two types of nucleic acids, right? That is DNA and RNA. DNA is the deoxyribose nucleic acids and RNA is the ribose nucleic acids. And here we have to know what are the components of the nucleic acids. So these are usually including one pentose sugar. If the pentose sugar is deoxyribose pentose sugar, that is called, nucleic acid is called DNA. If it is ribose sugar, that is called RNA. And both of them should contain a nitrogenous base. If it is DNA, it will be means there are again two types, pyrimidine and purine. So purines are of two types, adenine, guanine. Pyrimidines are of two types, uracil, thymine and cytosine. These purines and pyrimidines are usually classification is based on the structure of the corresponding nitrogen base. Okay. So the nucleotide is containing one more component, that is the phosphate group. So we have three components: pentose sugar, nitrogenous base and phosphate sugar. So based on this one, we can classify them into deoxyribose and ribose. So here we have the structures of the five different types of nitrogenous bases as well. So please remember the structures as well. Okay. And here we have the structure of DNA and RNA. DNA is double helical structured, RNA is single stranded. That is also important. So these are the important topics from the chapter Biomolecules. So now we will move, so these are the important topics. So now we will move to the structure, some important past paper questions from the chapter Biomolecules. Okay. So let us go through the first question. So the question is, here we have two statements. They are, first one, glycogen is similar to amylose in its structure. And statement two is, glycogen is found in yeast and fungi as well. So based on this statement, we have to find out the correct answer. Okay, so coming to the statements first. The first statement says that glycogen is similar to amylose in its structure. This is actually, glycogen means they are not similar to amylose. Glycogen is similar to amylopectin. Amylopectin. Okay, so the first statement given is wrong because glycogen is similar to amylopectin, not amylose. Okay, coming to the second one, glycogen is found in yeast and fungi also. So this we just discussed, right? So glycogen is commonly called in the commonly found in yeast and fungi. So the second statement is the correct one. So here correct answer will be option B. Because glycogen is actually a polysaccharide that serves as the main storage form of glucose in animals and fungi. And it is highly branched and primarily stored in the liver and muscles as well. And in the liver, glycogen helps to regulate the blood sugar levels, while in muscles, it provides the energy for the contraction. So when the body needs glucose, enzymes break down this glycogen into glucose 1-phosphate, which is then converted to glucose which is then converted to glucose 6-phosphate for use in metabolism. So the structure will be similar to amylopectin, and they can be also found in yeast and fungi as well. So the correct answer is option B. In the next question also we have two statements. In the first statement it says that a unit formed by the attachment of a base to one position of sugar is known as nucleoside. And the second one says that when nucleoside is linked to a phosphorus acid at fifth position of sugar moiety, we get nucleotide. So nucleotide and nucleoside are related to the nucleic acid, right? So here we have to know what is a nucleic acid and what are the components of the nucleic acid. This is what we discussed at the end of the topic. Nucleic acids are of two types. They are RNA and DNA. Okay. So if you consider the DNA or RNA, both of them will contain the components, that is the nitrogenous base or nitrogen base. Then it will contain a sugar, either ribose or deoxyribose sugar, then a phosphate group. Okay, and the nitrogenous base and the sugar moiety, that is the nitrogenous base and the sugar moiety together called nucleoside. Now, phosphate group when attached to the nucleoside, that is together called nucleotide. Means all these three together known as nucleotide, nitrogenous base and sugar is only called nucleoside. So here if you check the first one, a unit formed by the attachment of the base to position of the sugar is known as nucleoside. So that is the correct statement as we discussed now. And the second statement is when nucleoside is linked to the phosphorus acid at fifth position of the sugar moiety, we get nucleotide. Means the nucleoside when connected to the phosphate group, we will get nucleotide. That is correct. But in nucleic acids, the thing is, the nucleotide is formed by the combination of nucleoside with the phosphate group at the fifth position of the sugar molecule, not the phosphorus acid. So the second statement is the wrong one. Okay, so the correct answer will be option D. Now here we have to choose the incorrect statement regarding the enzymes. Okay, so first of all we have to know what are enzymes. So enzymes are the compounds which can seen in our body and they are called biological catalyst. Biological catalyst. Okay, so they are usually the catalysts. What the catalysts are doing, they can speed up the process, the chemical reactions, right? So similarly, the enzymes, which are biological catalysts, they can also speed up the processes that takes place in our body, in living organisms, without being consumed. And they are mostly proteins and work by lowering the activation energy of the reactions. So the biological catalysts or enzymes are usually proteins. Okay, and the enzymes are having specific active sites where that the mechanism of the enzymatic action we already discussed in the previous slide. So and we know that enzymes are having some specific active sites where the substrates can bind, right? So here we have discussed it, right? Yeah, so here we discussed it, that is when the enzymes are binding with the substrate molecules, they will produce the enzyme substrate complex, and after the reaction, the product will leave and the enzyme will be unaffected. So this is the reaction of the enzyme, right? So but there are some factors which can affect the enzymatic action, like temperature, pH, substrate concentration, and those kind of things. Okay, so based on these, let us check the options. The first option says that the like chemical catalyst enzymes reduces the activation energy of the biological process. So that is the correct one, because enzymes are biological catalysts, so they are reducing the activation energy of the reaction. Reducing the activation energy of reaction. So we have to choose the incorrect statement, right? So the first statement is the correct one. So that is not the correct answer. And coming to the second one, the enzymes are polysaccharides. That is not correct, because as I told you now, enzymes are proteins, not polysaccharides. So this is the incorrect statement regarding enzymes. So that is the correct answer here. And coming to the third option, enzymes are very specific for a particular reaction and substrate. Obviously, that is true. Coming to the last one, they are biological catalysts. That is also true. So the correct answer, which is the incorrect statement regarding enzyme, will be option B, means enzymes are polysaccharides. Next, next question. We have some diseases. We have some vitamins here. We have to find out the vitamin which is causing the RBC deficiency. So at the end of the slides, we discussed about the vitamins as well, and we know what are the different deficiency, different diseases arising due to the deficiency of different vitamins, right? So here we have the table. Vitamin A, B1, B2, all of them are causing different diseases. So here they are asking for the RBC deficiency. So RBC deficiency is causing due to the deficiency, RBC deficient in hemoglobin, that is pernicious anemia. Anemia is due to the deficiency of vitamin B12. So here the correct answer will be vitamin B12. Option B is the correct answer. Here we have four different amino acids here. We have to find out the one which is the basic amino acids. As we know, the amino acids are containing two groups, that is amino group and carboxylic group, right? So what is an basic amino acid? If the number of basic groups, that is the number of NH2 groups, is higher than that of number of carboxylic group, we can call it as a basic amino acid, right? So for the basic amino acid, which among the following is the basic amino acid? So the first one is tyrosine. So coming to the tyrosine, okay, coming to the tyrosine, which is a polar, slightly acidic amino acid, not basic. They are asking for the basic one, but tyrosine is slightly acidic in nature, and they are also polar. So tyrosine is not the correct answer. Next is lysine. Lysine is having amino group extra in the side chain which can accept a proton. So there are more amino group than that of carboxylic group, means lysine will be basic in nature. So that is the correct answer. If you check others as well, serine. Serine means this is a neutral amino acid, means they are containing equal number of CH group and NH2 group. Okay. So they are neutral. So this is again the incorrect answer. And coming to the last option, alanine. Alanine is also non-polar and they are also neutral. Non-polar, neutral. If you check the polarity of serine, that will be polar in nature but still neutral. So among the four options, the one which is a basic amino acid is lysine. Option B will be the correct answer. Coming to the next question, here we have some metal ions. It is given that the metal ions are activating many enzymes and they are participating in the oxidation of glucose to produce ATP, and with sodium that is responsible for the transmission of nerve signals. We have to identify the one which is activating the many enzymes, and which is many like which participate in the oxidation of glucose to produce ATP, and with sodium, which responds for the transmission of nerve signals as well. So from these options, the correct answer will be potassium, that is K, because potassium plays a crucial role for activating many enzymes involved in the oxidation of glucose to produce ATP, and it also aids in the transmission of nerve signals and working in conjunction with sodium. So the correct answer will be sodium, option. Sorry, so so the answer will be potassium, so option B. Now we have four different amino acids here. We have to identify the one which is the non-essential amino acid. So amino acids usually classified into essential amino acids and non-essential amino acids. The essential amino acids means they are not synthesized in our body, and we have to obtain it from our daily diet, or we have to get it from outside. So that is called essential amino acid. And non-essential amino acid means actually they are already in our body, so no need to get it from our food. So this is the difference between essential and non-essential. So here we have four options. We have to choose the one which is a non-essential amino acid. So first, let us see the first option, valine. Valine is actually an essential amino acid, means they cannot be synthesized in our body. So we have to obtain it through our food. So that is essential, which is the wrong answer. Coming to leucine, leucine is also an essential amino acid. So that is also an incorrect option. And coming to option C, alanine. This is a non-essential amino acids, means our body cannot synthesize this. So it does not need to be obtained. So it is need to be obtained through the daily diet. So this alanine is usually they can be synthesized in our body. So no need to get it through our dietary plan. So this is the non-essential amino acid. And means because this is the same reason they are synthesizing in our body itself. Lysine is also an essential one, means they are not synthesizing in our body, we need it from diet. But alanine means they are non-essential because it is already synthesizing in our body, no need to get from the diet plan. So that is the correct answer, option C. So the next question, we have to find out the difference between amylose and amylopectin. Amylose and amylopectin are actually two types of polysaccharides found in starch. Polysaccharides found in starch. Okay, so amylose is actually, if you coming to the structure of these two, amylose is a linear polymer of alpha D glucose, and they are linked by alpha. Amylose is linked by alpha 1,4 glycosidic linkage, and it forms a helical structure and is less soluble in water. Okay, and they are contributing to the form structure of the starchy foods as well. If you check amylopectin, it is a branched one. It is a branched polymer of alpha D glucose, and the linkage will be alpha 1,6. So in amylopectin, we can see alpha 1,6 linkage, and they will be more soluble and digestible than amylose. So this is the type of linkages present in amylose and amylopectin. Now let us see the options. Here it is given that amylose has 1,4 alpha linkage and 1,6 alpha linkage. This is not the correct answer, right? Okay, so coming to the options, the first option says that amylopectin has a 1,4 alpha linkage and 1,6 alpha linkage. This is the correct answer that we discussed now. All other options are the incorrect options. In a protein molecule, various amino acids are linked together by which type of linkage? So protein molecule is actually, it is a polymer of amino acid. Means various amino acids, means amino acids are the building blocks of proteins, means the monomers present in proteins are amino acids. And the amino acids in this protein molecule are linked together by peptide bond. That is, amino acid is containing both NH2 group and COOH group, right? So the hydrogen atom in the means one hydrogen atom from the amino group or hydroxyl group from the carboxylic group, they together remove as one water molecule, and there will be a linkage, that is NHCO bond between two different amino acid molecules. That is, the NH2 group of one amino acid interact with COOH group of another amino acid. That's why they are removing as a one water molecule, and they will produce and they will produce the CONH linkage, and that is called the peptide bond or peptide linkage. So that is the link or bond present between the amino acids. So glycosidic bonds are wrong answers, which are present between the monosaccharide units in a polysaccharide. Glycosidic linkage is not present in the protein molecule. So these A and B are wrong. Coming to the option C, peptide bond, this is the correct answer. So A and B are wrong, and dative bond is not the correct answer, because dative bond means it is a type of chemical bond, coordinate bond, which is produced by the sharing of electrons from only one atom. So that is not a kind of bond present between the amino acid residues. So the correct answer is peptide bond. Next question. The function of sodium pump is a biological process operating in each and every cell of animals. Which of the following biologically important ion is also a constituent of this pump? So sodium pump, or it is also called sodium potassium pump. So from the name itself, it is clear. So the potassium ions are contributing here. Before that, we have to know what is the sodium potassium pump, right? So this sodium potassium pump, or Na plus K plus pump, this is the active transport protein. That is the means the act. It is an active transport protein that maintains the electrochemical gradient in cells, and it moves three Na plus ions out and two K plus ions in the cell using ATP. This is called potassium sodium pump, and the function is, it is keeping the inside of the cell negative compared to the outside, and it helps to improve the, it helps in nerve impulse transmission and muscle contraction, and it is also maintaining the osmotic balance and cell volume. So these are the functions of, these are the important functions of this pump. So sodium and potassium ions are involving here. It means the correct answer will be option B. So go through them properly, then it will help you too much. Okay. So that's it. That's all about this class. So the next class we will meet with some other chapters as well. So bye bye. See you.